Hey guys, I'm back with another question. Today's question is discuss the role of phospholipids in maintaining the fluidity of plasma membrane. Okay, so basically how the plasma membrane works. That is what I'll have to discuss and what is the role of phospholipid in. So as we know, the current model that is accepted is the fluid mosaic model, and it was given by Singer and Nicholson. You know that. And what it states that the lipid part is fluid and the protein part is mosaic, and what is commonly said is protein icebergs in sea of lipids. Now let us see the basic structure. So this is how the bilayer is actually present. You already know that this is the tail part is the lipid part or non-polar part, and the head is the black circles represent the polar head or hydrophilic part, and the green part represents the hydrophobic non-polar tails, acyl CoA chains. Okay. Now this is the outer leaflet. Suppose, and this is the inner leaflet or cytosolic leaflet, right? So basically this violet part is the cytoplasm. Consider this as the endoplasmic reticulum. Then comes the nucleus, right? Okay, so this part is the cytoplasm and this part is extracellular. This is the basic structure, and there are several proteins are present, which may be peripheral proteins, which may be integral proteins. Okay, peripheral proteins are there, integral proteins are there, amphitropic proteins are there. That is not the topic of discussion here. We'll discuss it later if some question is present, right? So now coming to the next part, that why is the fluidity or how is the fluidity of plasma membrane maintained. So basically the fluidity is maintained by phospholipids, and the reason for fluidity is that the interaction between the lipid molecules, phospholipid molecules, is non-covalent, and because the interaction is non-covalent, the lipid or phospholipids are able to move freely within the lipid bilayer, and this basically accounts for the plasticity or fluidity of the membrane, right? Because the interaction is non-covalent. If it was covalent, then the movement would have been restricted, because covalent bonds are very difficult to break, and if all the molecules formed covalent bonds between them, then overall the structure will become very bulky, and therefore it will reduce the mobility. Now as you know, the plasma membrane basically exists in two states. Suppose this is the physiological temperature, right? T physiological. Okay, this is the physiological temperature. This is a temperature T1 which is lower than the physiological temperature, and there is a temperature T2 that is higher than the physiological temperature. The temperature increases in this order, right? Increase of temperature. Now I'm keeping this part the center. Now if the temperature is less than physiological temperature, then the plasma membrane exists in a liquid ordered state, right? Liquid ordered state or L0 state, where all motions that includes the lateral motion, right, in the same plane of lipid bilayer, all motion are constrained. Okay, all motion are constrained, and low fluidity is present in this state, right? And the membrane is less fluid comparatively of the fluidity that was present in the physiological temperature, and this is known as the liquid ordered state. Now if we keep on increasing the temperature, then the membrane will definitely be present in another state known as liquid disordered state, right? Liquid disordered state or the LD state, and here the motion increases. Overall fluidity or motion of the lipid molecules increases. Why it increases? There are two reasons. Number one, since heat is there, right? So because the temperature is high, the lateral diffusion, right? The lateral diffusion of lipid increases. Lateral diffusion of the phospholipids, I will write it as PL molecules, increases. And further, second reason is rotation about the carbon-carbon bond of long acyl chains, the hydrophobic part, right? Long acyl chains also increases. Therefore, in liquid disordered state, the fluidity increases, and liquid ordered state, the fluidity decreases. The temperatures that would require to convert liquid ordered state to liquid disordered state is known as transition temperature or melting temperature. Please note that in both of the states, right, the stability and structure or dimensions are maintained. Dimensions are maintained. Only there is an increase in fluidity, but the stability, dimensions, all of that properties of the membrane are maintained so that it is able to work efficiently. Right, now coming to the role of phospholipids. So mainly three broad types of phospholipids are present, and the first one is, okay, I will do it in a table format, right? Okay, so the first type of lipid that is present is known as saturated phospholipid. The second is unsaturated, means a double bond is present. And the third type is, this is not that important, that phospholipids with short acyl chains. Right, short acyl chains basically means smaller phospholipids, right? Now let us see how they affect the fluidity. See, the saturated phospholipids, if are present, will be present very close to each other, right? They will be able to form a tight interaction or tightly interact with each other because the hydrophobic part, the chains or acyl chains of this saturated phospholipids are straight, right? And therefore they can easily pack together, right? So therefore, if in a membrane the saturated phospholipid is high, then the fluidity will be low, fluidity decreases with increase in saturated phospholipid. Now coming to the unsaturated phospholipids, these unsaturated phospholipids have a unique type of structure, right? There is one normal acyl chain, and the other part, or one phospholipid, or both, may have a double bond, and due to this double bond there is a kink produced, right? This type of bend is produced. Usually it is a cis configuration, right? Cis configuration of phospholipids usually increase the fluidity, right? And since there is this kink produced, now the second phospholipid molecule cannot pack closely, right? It has to be at a distance. And there is another kink, like this. Hope you get the point, that because of this kink produced, the phospholipids will be present at a distance apart from each other instead of being closely packed as in saturated phospholipid. So as you see, the space increases, and therefore this also increases the fluidity. So fluidity increases with increase in unsaturated phospholipids. And lastly, short acyl chains. So suppose this is the bilayer again, right? And these are some of them have very short acyl chains. So these phospholipid molecules with short acyl chains will be able to move faster, right? Because they are short, their weight is also less, right? So therefore mobility is faster, and therefore there will be increase in fluidity. In phospholipids, increase in phospholipids with shorter acyl chains. So this is how the phospholipids actually maintain or regulate the fluidity. For example, if a bacteria is you are doing a culture of a bacteria, right? Bacteria is being grown on a culture medium, and if the temperature is very low, right, low temperature, then at low temperatures the fluidity should decrease. So to maintain fluidity, what the bacteria will do, it will increase the synthesis of unsaturated phospholipids. So as we already know, if the unsaturated phospholipids increase, it will also increase the fluidity of the membrane. Okay, so hope I was able to explain the concept. There is also a role of cholesterol in maintaining the fluidity of this plasma membrane, but it is not the topic of discussion for this video. Hope I was able to explain properly. If there are any mistakes in this video that you find, you be sure to let me know in the comment section below. And if you like the video, like it, share it with your friends, and make sure to subscribe to my channel. Thank you for watching.